Good evening to everybody. Uh, Welcome back to again Adya online biotechnology classes. Today I am going to explain about central dogma. So everybody confusion in this topic. What is a central dogma? The central dogma topic, it is a very important for CSIR net and JRF or APC. So first of all we need to understand what is the central dogma of molecular biology or central dogma of molecular life. So first of all, central dogma of molecular biology, it means by the process of transcription, by the process of translation, and by the process of reverse transcription, called as a central dogma of molecular biology or central dogma of molecular life. It is a main main for the central dogma of molecular biology. The passage of the information transfer from one generation to another generation and one bacterium cell to another bacterium cell, called as the central dogma of molecular biology or central dogma of molecular life. So first of all, what is a transcription? Okay, so what is central dogma of molecular biology? By the process called transcription, translation, by the process called translation and reverse transcription. Okay, reverse transcription process. So first of all, what is the transcription? Synthesis of mRNA. Okay, synthesis of mRNA process called transcription. And what is the translation? Synthesis of protein called as translation. Okay, synthesis of protein called as translation. And what is the reverse transcription? Synthesis of cDNA called as complementary DNA by the process called reverse transcription. Okay, so what is central dogma of molecular biology? The passage of information from the DNA to mRNA, mRNA to protein, called as central dogma of molecular biology by the process called transcription, translation and reverse transcription. We need to understand what is the transcription? Synthesis of mRNA. What is the translation? Synthesis of protein. What is the reverse transcription? Synthesis of cDNA, called as reverse transcription. So this whole process is also called as central dogma of molecular biology. Okay, central dogma of molecular biology. So first of all, who discovered by the double helical structure of the DNA? So double helical structure of the DNA was first proposed by the James Watson. Okay, James Watson and Francis Crick. Okay, James Watson and Francis Crick in the year 1953. Okay, in the year 1953. Who discovered by the double helical structure of the DNA? James Watson and Francis Crick in the year 1953. Okay, so later, so the before Watson and Crick, the other scientist is Paul. Okay, other scientist is Paul. So before Watson and Crick, so another scientist is named as Paul. Who is Paul? He has identified as triple helical model of the DNA. Okay, triple helical model of the DNA. Okay, triple helical model of the DNA. So Watson and Crick is first discovered by the double helical model of the DNA. Okay, double helical of the DNA. So the double helical model of the DNA was first proposed by the Watson and Crick. Okay, so Watson and Crick in the year 1953. Okay, in the year 1953. Before Watson and Crick, another scientist is Paul. Who is Paul? Paul is he has identified as triple helical model of the DNA. Okay, triple helical model of structure of the DNA. So this triple helical structure of the DNA, the most of the scientists is oppose this particular statement because the reason is there is no any structural identification proof and there is no any experimental evidence. So the triple helical model is totally wrong. Okay, so totally wrong. So and later and later the students of Watson and Crick, the students of Watson and Crick. So these Watson and Crick is proposed by the double helical structure of the DNA. It is under the influence of X-ray crystallography. Okay, X-ray crystallography, X-ray crystallography structure. Okay, X-ray crystallography structure. So next, what is the main function of the DNA? Okay, so DNA the main function any? The main function of the DNA, synthesis of protein. Okay, synthesis of protein. The main function of the DNA. So function of DNA, synthesis of protein. So what is the main function of the protein? Protein's main function. So protein's main function, the 37.2 trillions. 37.2 trillion, 37.2 trillions of cells are present in our body. So every single cell or they will required, okay, they will require the proteins, they will require the protein. Example, the main type of proteins is hemoglobin, and another one is actin and myosin, and the third one is antibody. Okay, third one is antibody. So first of all we will discuss each and every step. So first of all, hemoglobin. So what is the hemoglobin? Hemoglobin is a special type of protein is present in inside the our blood. Okay, what is the main role of hemoglobin? Hemoglobin, they transport the oxygen. Okay, they transport the oxygen in the blood. So hemoglobin main function, the transport the oxygen in the blood. Okay, next one is the actin and myosin. So actin and myosin is a play role in contraction, relaxation. Okay, if contraction, relaxation, the muscle contraction and relaxation. If the muscle will be contraction, actin is present, and if the muscle will be relaxation, myosin protein will be present. Okay, so the actin and myosin is a main important role is the contraction of muscle cells, okay, contraction of muscle cell. So next, last one is the antibody. What is the antibody? So if there is a antigen, so if there is a antigen is present or enter into our body, okay, if the antigen is enter into our body, so what will happen? The antigen are going to synthesize antibody. So this antibody is a type of glycoprotein. Okay, is a type of glycoprotein. So what is the hemoglobin? Hemoglobin is a special type of protein is present inside our body, is present in the blood cell. So it is required for the transportation of oxygen, and actin and myosin is a play role in contraction of muscle cells. So what is the antibody? Whenever antigen is enter into our body, so these antigen are going to synthesize and a special type of antibody molecule. Antibody is nothing but glycoprotein. Okay, antibody is nothing but glycoprotein. So every single cell is performed by the living cell, and these living cells are under controlled by the protein. So thus that particular type of proteins are going to synthesize a unique molecule of DNA. Okay, unique molecule of DNA. So we need to understand what is the central dogma of molecular biology or central dogma of molecular life. So first of all, so this is the double-stranded DNA. So this is the double-stranded DNA molecule, and these DNA are going to synthesize mRNA. And these mRNA are going to synthesize protein molecules. Okay, are going to synthesize protein molecules. These are the proteins. Okay, these are the protein. So first of all, what is the central dogma? By the process called transcription, translation translation and reverse transcription. Okay, reverse transcription. Okay, by the process called transcription, translation and reverse transcription. This whole process is also called as the central dogma of molecular biology. What is the transcription? Synthesis of mRNA. What is the transcription? Synthesis of mRNA from the DNA molecule. That is called transcription. What is protein? All amino acids are protein, but all proteins are not amino acids. Why they? Because all amino acids are linked with peptide bond to form a polypeptide chain, gets converted into proteins called as proteins. Okay, so first of all, so this type of DNA, okay, DNA is not going to synthesize, is not going to synthesize direct synthesis of protein. So DNA is not directly synthesis of protein. Okay, so direct synthesis of protein. Why does this the RNA molecule? Why does this RNA the DNA? So DNA is going to synthesize mRNA the instead of RNA polymerase enzyme. RNA polymerase enzyme. So the DNA is not directly synthesis of protein molecule. Okay, protein molecule. So first of all, so before the transcription, so this DNA it undergoes replication process. Okay, DNA it undergoes replication process. So first of all, transcription, replication, translation. So this reactions is catalyzed by the three type of enzymes. One is the DNA dependent RNA polymerase and DNA dependent RNA dependent DNA polymerase and reverse transcriptase. Okay, reverse transcriptase. Okay, so these transcription, translation, reverse transcription under controlled by the, it is catalyzed by the three type of enzymes will be there: DNA dependent RNA dependent DNA polymerase and reverse transcriptase enzyme. So first of all, the before the transcription this DNA is undergoes the replication process. Why they? Because the unfavorable condition, the DNA get damaged. So these damaged DNA are not going to synthesize protein. So the DNA molecule before the transcription, DNA it undergoes replication process. What is the replication? The multiple copies of its own DNA. And Xerox copies of its own DNA. Duplication process. Okay, the DNA it undergoes replication process. To form new DNAs or daughter DNAs will be formed. Okay, daughter DNAs will be formed, and this replication takes place in the S phase of the cell cycle. It is the most important bit for MSc purpose. So replication takes place in the S phase of the cell cycle. So this DNA it undergoes replication process to form daughter DNAs. So which is catalyzed by the enzyme? RNA dependent DNA polymerase. Okay, RNA dependent DNA polymerase, or otherwise known as DNA dependent DNA polymerase. So DNA dependent DNA polymerase is a main important enzyme is present in the DNA replication process. So the DNA it undergoes replication process to form daughter DNAs. So by the process called replication, this replication takes place in the S phase of the cell cycle, which is catalyzed by the enzyme DNA dependent DNA polymerase. Next, transcription. So what is the transcription? So the transcription means the ribonucleic acid which is synthesized from the DNA called as transcription. Okay, what is the transcription? Synthesis of mRNA. So synthesis of mRNA from the DNA molecule called as transcription. So in this transcription, which is catalyzed by the enzyme DNA dependent RNA polymerase. Okay, DNA dependent RNA polymerase. So DNA dependent RNA polymerase. Okay, so sorry, RNA dependent DNA dependent RNA polymerase. Okay, so when the DNA when the DNA it undergoes transcription process to form mRNA. Okay, so these mRNA comes and binds with the ribosomes. mRNA comes and binds with the ribosome. So these ribosomes, it is a ribosomes is a okay, right, the mRNA comes and binds with the ribosome. So ribosomes is a main for protein synthesis. The ribosomes comes and binds with the mRNA, so gets converted into proteins under the translation process. Okay, translation process, the ribosomes comes and binds with the mRNA, it undergoes translation process to form proteins. Okay, next, what is reverse transcription? The mRNA, so the mRNA gets converted into DNA. The mRNA gets converted into DNA by the process called reverse transcription. Reverse transcription in the presence of enzyme. In the presence of enzyme, RNA dependent DNA, sorry, reverse transcriptase enzyme. Okay, reverse transcriptase enzyme. So if you everyone, if you want any doubts, I will clarify with them. Okay, so this is about central dogma of molecular biology. So one the DNA are not going to synthesize protein. Why they? Because the DNA is a highly stable molecule. Highly stable molecule, the DNA to protect its integrity, and the DNA is protect stability. Okay, and its stability. So these DNA are going to synthesize, are not going to synthesize proteins. Okay, are not going to synthesize protein. So the instead of RNA polymerase, the RNA polymerase, it is catalyzed RNA polymerase, it is undergoes the transcription process.